Hello, my name is Anar, and I work at the Leonard Davis School of Gerontology at the University of Southern California. And here I'm presenting on the brain age and how to measure it experimentally. So a prerequisite for this tutorial will be the one on brain structure and how it changes in old age, given by my colleagues Nahyan and Heel. So here we go. So in humans and in many other species, I presume the brain aging or aging of the human brain is accompanied by cortical thinning. The cortical layer of gray matter becomes thinner and thinner, loses some of its volume, some of its thickness as we age, and it becomes particularly noticeable as we get very old. So you've seen some of that from the presentation by Nakian and Heel. And here I'm showing just a sort of a cortical overlay figure or figure on the brain surface of the sample cortical thickness, at least the cortical thickness change as it goes on in aging humans. So a quantity like this measured on the entire cortex for the entire brain will be useful in measuring or quantifying the brain age for us. So what is brain age and how does it work? So we will show how brain age works and how it's computed on an example of cortical thickness measurement, and we'll use it as the quantity that gives us the brain age. So brain age is essentially an indication of how fast we are aging. And brain age can equal the chronological age, but in general, it's not the same as your chronological age. The chronological age being the number of years you've lived, essentially, since you were born, calendar years. Now, brain age equals, by definition, chronological age, but only if brain of the person has aged as expected for that chronological age. So how do we compute it? So it's essentially, we're trying to capture this cortical thinning process into a measure of aging sort of instead, and we call that brain age. So we try to, given the data in the blue points over here, we try to, the data is something that might be expected from a healthy or typically aging population. And from them, we try to quantify this function that is indication of exactly how you're expected to age on average in terms of your cortical thickness. So you start out here, you're very young, you have a very thick cortex, and then people move along this trajectory as time goes on, their cortex become thinner and they themselves become chronologically older. So if you're staying on this line, it means that you're aging exactly as expected and your brain age is exactly the same as your chronological age. Now, let's take an example. Here in the black rectangle here, we have a data. This data just gives us the essentially the cortical thickness of the person. And this person's cortical thickness happens to be very low. It's a very thin cortex. And we're asked the question of, okay, so what is their expected chronological age of this person given their cortical thickness? And we might learn that if we were as exactly expected from the population on average, we would get in a chronological age like this, a very old person, because their cortex is very thin. Now, what happens if we have a new data? A new data that, let's say, has both in the green triangle, both the cortical thickness value and the chronological age value. Now, from this two data points, two values, we want to decide if the person is aging as expected or not as expected, either faster or slower. So from the fact that the green triangle is not on this blue line, we can tell that they're not aging as expected. But are they aging faster or slower? So let's look at the point. It looks like for their cortical thickness, the vertical line, let's say, imagined here, their chronological age is much larger. So they're above this line in terms of age, chronological age. So that means they're chronologically, they're very old or older than expected, but their brain, based on cortical thickness, is younger. So that means it looks like they're aging well, in the sense that aging in a way such that their brain is actually not losing their thickness as fast as it could have. So that's a good thing. And how about, so here's the indication that says for their particular chronological age, uh, their cortical thickness, let's say, their cortical thickness is shown on the horizontal axis, and their chronological age on the vertical axis is much larger than their expected chronological age, which is the green dashed line. So they're much younger in terms of brain cortex thickness than they're expected. How about this data point? So the red diamond here is below the line. Already the fact that it's on the other side of the expected line might tell that it's a bad thing, but let's just verify. So for their particular cortical thickness, the age that we expect it to be at, the chronological age, is this value, the red dashed line, but their real chronological age is much younger. So this tells us that their brain age, or the expected chronological age from the thickness, is something larger above here than their real chronological age over here. So that means their brain has sort of accelerated its aging. Now, how about if we have points like this? So there are a bunch of points here, and this is another population. This population might be either some very healthy aging people or some other people. We don't know exactly what this group is, but we want to see that there's some difference between the blue group, the expected, or the sort of the very typically aging group. Now, in general, what's happening is that the red diamond points are below the chronological age of the blue line, and they're to the left, or they're thinner than the blue line. So they're both younger than the expected chronological age for their thickness, and they're thinner in cortical thickness than expected for their chronological age. So it looks like they're the population that's aging not so well, in the sense that their aging is accelerated, and they might be a group that's not so good. So let's verify that. Uh, for each horizontal line here, for a particular chronological age, we would expect a typically aging person to be on the blue line, which is let's say right over here, and their cortex to be this thick. But the red points are to the left, or their cortex is thinner. So their cortex has became thinner for the same chronological age. Now, also this indicates that their cortical thinning is accelerated, or same cortical thickness. Let's say this particular cortical thickness is reached at a much younger chronological age, much below than the blue points, the expected or the typical points here. So the typical brains have larger chronological age for the same thickness compared to our red points. So the people in red points are younger, and they've already lost cortical thickness to the degree that people at a much older age do. So this is not a good thing. And potentially this group is maybe an Alzheimer's group. And as part of the disease process, they might be losing cortical thickness, which is a known process in humans. So that is the sort of summary of how brain age works and what it is and how it's computed. Brain age is essentially the expected chronological age for a particular cortical thickness in our case, or some other measure. So it can be brain age computed from other quantities, like could be the cortical volume or some other quantity. But in our simplified case of one variable, we're just measuring brain age as the expected chronological age of the person given their cortical thickness. Okay, so there will be a few questions on the quiz on the topic of brain age, and we will finish here. Thank you.